All right, so now we'll look at the part 2 of the cell. Our focus for this discussion is on the cytoplasm. The cytoplasm is divided into two parts. Cytoplasm is divided into two parts, which is cytosol and organelles. Apart from that, we have what we call the cytoskeleton. So, pardon me, cytoplasm is divided into three parts. Cytosol, which is a fluid part of the cell. Then we have cellular organelles. Then we have the cytoskeleton. So three parts in total. So organelles, by definition, are metabolically active structures. And there are two types of organelles inside the cytoplasm. There are membrane-bound organelles and non-membrane-bound organelles. So what do we mean by membrane-bound? These are organelles surrounded by a membrane similar to the cell membrane in structure. So I'll refer you to part 1 where we discuss the cell membrane. Then we have other organelles that do not have a membrane around them. So we call them non-membrane bound organelles. So as we discuss these organelles, we'll see which ones are membrane bound and which ones are not. So since the cytosol is just a fluid part which provides an aqueous environment in which various chemical reactions take place, we are not going to discuss it here because it's just the aqueous environment. Our focus will be on organelles and the cytoskeleton. So let's begin with the largest organelle of the cell, perhaps the most important, which is the nucleus. So the nucleus is a membrane-bound organelle. It is surrounded by a membrane. Now, apart from that, the nucleus has two membranes. Cell membrane is a single membrane, which is formed of a lipid bilayer, but the nucleus has two membranes. So we call them nuclear envelopes. Inner nuclear membrane, outer nuclear membrane, or if you like, inner nuclear envelope and outer nuclear envelope. I'll use a structure for easier explanation. So what we are seeing here, if you can see the pointer there, is the nuclear membrane. Now observe that the nuclear membrane has two membranes. This will be the inner membrane or envelope. That will be the outer membrane or envelope. That being said, meaning there's a small or minute space between the two membranes. There's a small or minute space between the two membranes, which you call the perinuclear space. Perinuclear space in here, meaning substances that will leave like that or enter the cell have to pass through two layers of membrane, the outer and the inner, and reach the inner side of the nucleus. The space inside the nucleus is called the nucleoplasm. Nucleoplasm. We see here it's appearing grayish. Then inside the nucleus or inside the nucleoplasm, we have what we call the nucleolus. Nucleolus is a small particle within the nucleus composed of RNA. So this structure we're seeing here, appearing brownish, is a nucleolus, and this is where ribosomes are produced. So it can be considered a small organelle within a nucleus, nucleolus. Then the entire nucleoplasm is filled now with DNA. So the nucleolus, you find RNA there. The whole nucleoplasm is filled with chromatin. Now, this chromatin is simply DNA, what we call chromosomes. Now, we only call them chromosomes when the cell is about to divide. When the cell is in its resting stage like this, where it is just performing various functions of the body, we call it chromatin. When this cell is about to divide, these chromatin are coiled to form what we call chromosomes. So if I can simplify it for you, I'll just say chromatin is uncoiled DNA, whereas chromosomes is coiled DNA. That being said, I want us to observe another special thing about the nucleus. We see that in the nucleus, there are these small perforations connecting the external environment of the nucleus to the internal environment of the nucleus. Remember that outside the nucleus is cytoplasm. Inside the nucleus is nucleoplasm. So we call these small perforations as nuclear pore. Nuclear pores. So these nuclear pores are essential for transportation of ribosomes, since we are saying the nucleolus is an organelle that produces ribosomes. Ribosomes have to pass through these pores to go into the cytoplasm. They have to leave the nucleus because ribosomes perform their functions in the cytoplasm. That being said, since the entire nucleoplasm is filled with DNA in form of chromatin, and DNA is a molecule of life, all information about us as human beings is contained in our DNA. Therefore, the nucleus and its main function is to control all cellular activity, and at the same time, produce ribosomes from a small, dense region found within the nucleus known as nucleolus. Two functions they are mentioned. This is a nucleus shown there, and this is a whole cell. That is a cell membrane here. And this is a nucleus, and that is a nucleolus. So the nucleus is the largest organelle, like we have said, and all these I've already explained. Ribosomes are produced in the nucleolus. Let's look at ribosomes then. What are ribosomes? So ribosomes are minute organelles, very small indeed. Organelles found in the cytoplasm, but they are produced in the nucleus. So ribosomes have two parts. Ribosomes are made up of two parts, shown here in the diagram. A large subunit, that is a large subunit, and a smaller subunit. So we can liken the ribosome to two rocks. A small rock on top, a big rock on top of a smaller one. More like an improper fraction, if you like. Large subunit above, small subunit below. So these parts, the subunit above, the larger one, and the small subunit below, are produced separately, and then they migrate from the nucleus here and pass through the nuclear pore and reach into the cytoplasm as single units. Then they assemble to become one organelle within the cytoplasm. And now we can call it a ribosome. What is the function of a ribosome? Ribosomes are the only organelles in the cell that will produce proteins. So their major function is protein synthesis. Like we can see here, these are amino acids. Amino acid, amino acid, amino acid. Then this is what we call transporter RNA. Then that one is messenger RNA. So of all organelles in the cell, it's only the ribosome that has a receptor for messenger RNA. See where the arrow is pointing? Messenger RNA come from DNA, which is inside the nucleus. It comes out like that and give us this molecule. It is messenger RNA that carries information about what type of protein the ribosome should produce. And that's a major function of the ribosome, is to produce proteins. Okay. Now, there are two types of proteins produced in the body. Those produced by ribosomes that are free, and those produced by ribosomes attached to the rough endoplasmic reticulum. Two types of proteins are produced by the cell. Proteins produced by ribosomes that are free, and protein produced by ribosomes attached to endoplasmic reticulum, making this rough endoplasmic reticulum. So where do proteins produced by free ribosomes go? So these proteins produced by free ribosomes work within the cell. They work within the cell. They'll either help us maintain the mitochondria, form peroxisomes, and also repair the nucleus. But proteins produced by ribosomes attached to endoplasmic reticulum are destined to work in faraway tissue, and therefore they'll be secreted from the cell. Some of them will remain and replace some damaged cells, some damaged proteins within the cell membrane. So in short, proteins produced by ribosomes on rough endoplasmic reticulum are for external use. Proteins produced by ribosomes that are free are for internal use, summarized as such. Okay, we look at another organelle. Before I go on further, a ribosome is not membrane bound. So this is an example of a non-membrane bound organelle. It has no membrane around it. So let's look at the endoplasmic reticulum itself. So endoplasmic reticulum are of two types. There's what we call rough and smooth. So in summary, the smooth endoplasmic reticulum is where lipids are synthesized, and rough endoplasmic reticulum is where proteins are synthesized, but several other functions come in. Now, let's start with the rough endoplasmic reticulum. We have already discussed part of it under ribosome. So I want us to focus on this diagram. This is a nucleus, nucleolus inside there. Then whatever we are seeing there is chromatin, those strands inside the nucleoplasm. Now, let's observe that those yellow spots we are seeing there on the surface of the nucleus are the ribosomes that are produced from the nucleus. They live through the nuclear pore and go and attach to the endoplasmic reticulum, converting it into rough endoplasmic reticulum. So we see that the nucleus is always surrounded by rough endoplasmic reticulum. Then peripheral to the rough endoplasmic reticulum is the smooth endoplasmic reticulum. So what is this organelle? This organelle is nothing but tunnels in which various chemicals are produced and processed. You see that's a tunnel, that's a tunnel. Then even here is a tunnel. Okay. So that is a rough endoplasmic reticulum. So apart from producing protein, the rough endoplasmic reticulum modifies protein by various processes, which we call, for example, sulfation, which is addition of sulfur, folding, which is compression of the protein that is formed, then glycosylation, which is addition of glucose to the produced proteins. So the moment you add sulfur, the property of that protein changes and its function changes. Same with a compressed protein and a glycosylated protein. And out of that, now you're going to have different types of proteins. For example, some are enzymes, some are hormones, just by addition of these various chemicals to them. Okay. Now, we said the space between two nuclei, between two membranes of the nucleus is a perinuclear space. Now, what about the space between the nucleus and the rough endoplasmic reticulum? We call that as a perinuclear cisternae. You see that this is a nucleus. There's a small space there in between the nucleus and the rough endoplasmic reticulum. This is called perinuclear cisterna. So they are not in physical contact. Yes, the nucleus is surrounded by endoplasmic reticulum, but there's no contact between the two. There is always a space, which we call the perinuclear cisterna. What about rough endoplasmic reticulum? It's also a tunnel. You can see that. So this organelle, smooth and rough, is one, except one has ribosomes on its surface. And both are membrane bound. You can see that is membrane. This is membrane. So they are membrane bound organelles. Okay. Function of the smooth, of course, is production of lipids or protein, lipids or fat, if you like. Apart from that, the smooth endoplasmic reticulum is an organelle that performs detoxification. So removal of harmful substances from the cell is a reserve of the smooth endoplasmic reticulum. Example given, alcohol. Alcohol has no nutritional value to the body. Even though we consume it, it contributes nothing to the survival of cells. If anything, it causes harm. Barbiturates, these are drugs. They also cause serious harm to smooth endoplasmic reticulum. So since these are toxins, the smooth ER must detoxify them, and then the kidneys will do their job by removing these harmful substances from tissues and blood. Now, let's observe that in muscle cells, specifically muscle cells, skeletal muscles, the function of the smooth endoplasmic reticulum is different. The smooth ER in muscles, specifically skeletal ones, is to store calcium ions. So all those functions of detoxification are reduced so that this organelle this time can store calcium. So in muscle cells, skeletal muscles, this will be full of calcium. And we know that calcium is essential for muscle contraction. And since the body does not produce calcium, we need calcium from diet. The muscle cells have adapted by replacing the functions of the smooth ER of detoxification and production of lipid to storage of calcium, which you can also call sequestration of calcium. If calcium levels drop in the smooth ER, then what follows next? You see tetani, muscle spasms, tremors, because muscles will start relaxing and contracting without control. You see that the eyelid, either upper or lower lid, begins to blink without your will. And sometimes we say, no, I'm expecting some good stuff. It's not true. Actually, there's just low calcium in your body. And also remember the hormone responsible for calcium levels. We are talking about calcitonin and parathyroid hormone. Those two hormones work in opposition to control calcium levels together with the kidneys. Now, let's look at other organelles. We look at the Golgi apparatus. Golgi apparatus, or otherwise known as Golgi body, Golgi complex. It is also another organelle that is membrane bound. So it is surrounded by a membrane. So what happens here is that the Golgi apparatus is slightly different from the smooth ER in the sense that it is also a series of tunnels, but these tunnels are flat. They are not tubular like we saw in the rough and smooth ER. And because of that variation from the ER, the Golgi apparatus is specialized for, number one, packaging. So substances will be produced by rough or smooth ER and enter this system or organelle. And while they are passing through these tunnels, those substances are sorted out, further processed, if you like, some modified. Once that is done, the Golgi apparatus will now store that substance as separate substances from one another and form these minute structures we are seeing here, which we call vesicles. These vesicles are for easier transportation because the cytoplasm, there are so many substances and molecules in there. It will be difficult, for example, if an enzyme is formed, to negotiate through the cytosol and leave the cell and go where it is needed. For example, clotting factors that we need in blood, hormones that we need to be produced from the pancreas go into blood and influence glucose, like insulin, glucagon. So the only way to protect them is put them in a certain package, and that is a function of the Golgi apparatus, is to form these small transport vesicles or vacuole, if you like. You see that the vesicles are detaching. You see here it swells a bit, detaches, begins like that, grows, finally separates. Now, because of this, the Golgi apparatus has to be, the membrane of the Golgi apparatus has to be continuously be replaced by a new membrane. And therefore, if that doesn't occur, you find that this organelle may finish because this membrane is detaching. So it requires constant repair. Apart from that, the Golgi apparatus is the main organelle in the cell that will now produce carbohydrates needed for that cell. So packaging, formation of vesicles, and production of carbohydrates. Carbohydrate is essential to give us energy. So just like the way the whole body needs energy, individual cells also require energy. Now, once carbohydrate is produced, in its state like that, it's useless to the body. Carbohydrate has to undergo several reaction processes in which oxygen is added to it so that we can now produce a cellular form of energy known as adenosine triphosphate. And it's only the mitochondria that can do that. And that now brings us to another organelle known as mitochondria. Mitochondria is rod-shaped. This is mitochondria here. This is mitochondria as well. Now, mitochondria, just like the nucleus, has two membranes also. So it is one of those organelles that are membrane bound. It has outer membrane and inner membrane. Now let's observe the inner membrane. The inner membrane has folds. You see the inner membrane goes like that, then folds on itself, back, folds, folds. So these folds are what we call cristae. The folds are what we call cristae. Then the space between the outer membrane and inner membrane is what we call the matrix. Matrix is space between the two membranes. Then the folds are what we call cristae. Now, mitochondria, the number of cristae it has is equivalent to the functional capacity of that mitochondria. So the less active the cell is, meaning its mitochondria has little number of cristae or folds. For example, bone cells, cells of cartilage, those cells are not so active metabolic-wise in terms of function. So they have little amount of cristae. Now, if you look at muscle cells, cardiac muscle, skeletal muscle, smooth muscle, these muscles are always functional every day. Nerve cells, cells that do not rest, they are always functioning. They have higher number of these folds or cristae. Then, apart from having higher number of cristae, cells who have higher functional capacity like nerves, muscle cells, they also have higher number of mitochondria. And those cells that are inactive also have little number of mitochondria. So, how does mitochondria produce energy? It can be summarized in this process. Okay, there's what we call glycolysis and citric acid cycle. Okay, the product of glycolysis is pyruvate here. Glucose, remember where glucose was coming from? The Golgi apparatus. Then it is transported to the mitochondria. Glucose is broken down to give us ATP. So that process is known as glycolysis. Okay. Then the product is pyruvate. Pyruvate enters the mitochondria. So this ATP has gone in the cytoplasm. Pyruvate further is catabolized by so many enzymes. One of them is NADH, which is seen here as well. Then enters another cycle, which is known as citric acid cycle. Now we see that citric acid cycle also gives us ATP. Aside from that, it will give us water as another product. So at the end of the day, when sugar is broken down in the cell, what we are looking for is ATP, which is energy for the cell, and finally water, because oxygen was added to sugar. This is pyruvate, then oxygen is added. Here the process known as oxidation. Oxygen is coming from red blood cells, enters in cells, enters in mitochondria, then added to this process to give us now ATP plus water. And this organelle is now known as the powerhouse, because without it, we don't have this form of energy known as adenosine triphosphate. Aside from that, mitochondria also has its own DNA. So we saw that DNA was inside the nucleus, but see, there's also DNA in the mitochondria. Now let's look at the nucleus. I'll take you back a bit. This is the nucleus, and this is DNA. Now, this DNA found in the nucleus is different from DNA found in the mitochondria simply because DNA found inside the nucleus is helical or spiral. You see that if you follow this, it forms a spiral. It is spiralating like this, going in a S-shaped manner. But DNA found in the mitochondria is circular. It's like a circle, like this. It forms like a circle. So there are two different types of DNA. Okay. So why is this organelle having DNA? It's for a simple reason, because mitochondria are self-replicating organelles. They can multiply in number if the demand of energy in that cell increases. For example, this person starts to work out. You start going to the gym, you start running, cycling, running long distances or sprints, such. You find that the energy demand for your muscle cells will increase, and the number of mitochondria in those cells needs to increase also, so that it can give you the energy that you need. And how will this organelle multiply itself? It's because it has DNA in it. And that's why the way mitochondria divides is similar to the way bacteria divides, which you call, the process is known as binary fission. So what happens is this organelle will swell or increase in size, hypertrophy, then split in half. As the cell is swelling, even the DNA is replicating, is increasing in number. So you're going to have two circular DNA. And when the cell divides, one DNA goes to the other cell, the other into another cell. And that way the number of mitochondria increases. One last point about mitochondria is this. The mitochondria that we have in our cell are said to be maternal in origin, meaning mitochondria comes from our female parents. So during fertilization, when the sperm is entering into the ovum, by the way, in number or in size, the ovum is way big compared to the sperm. The sperm is too small. So when the sperm is entering the ovum, the tail of the sperm remains outside. It's only the head that goes in. So when the tail remains outside, all the mitochondria for the sperm cell are located in the tail region. So they don't become part of the zygote, meaning the only mitochondria in the zygote is coming from the ovum. And thereafter, mitochondria begins to increase in number as the zygote keeps on dividing into a morula, a blastula, you know those stages, and we'll cover them later on. And therefore, some people suggest to say intelligence is from our mothers, not our fathers, because for the nerve cells that we use to think, they are depending on the ability of the mitochondria to power them so that you can think deeper. So thinking depends on mitochondria. Mitochondria comes from our mother, and therefore intelligence is linked to our mothers strongly than our fathers. That's all about mitochondria. Now, let's look at another organelle. We look at peroxisomes. So peroxisomes are also membrane bound. They are surrounded by a membrane. Now, these organelles are very small. You can see them here. Peroxisome, peroxisome, peroxisome. If I take you to this diagram, we can also identify them here. These are peroxisomes. Peroxisome, peroxisome. What are they doing in the cell? So peroxisome have oxidative enzymes. Okay, so these enzymes help in processing oxygen. Okay, so they are oxidative enzymes and therefore are involved in various metabolic reactions of the cell. Okay, so they are present in almost all human cells. And we're saying they give enzymes, and you know that enzymes speed up chemical reactions. So for example, for us to speed up the process of formation of ATP, we need enzymes involved, like I showed NADH. Peroxisomes are providing those enzymes. What about lysosomes? Lysosomes are another organelles that are bound by membrane, meaning they are surrounded by a membrane. So lysosomes can be located here in this structure. We see lysosomes here, lysosomes. Now, all cells need to defend themselves, whether they are white blood cells, red blood cells, and like they all have the ability to protect themselves to some extent, except that white blood cells have more developed lysosomes and therefore can identify and kill off so many microorganisms. Now, lysosomes produce enzymes. Now, this time, these enzymes are different from what peroxisomes are producing. These are hydrolytic enzymes. Enzymes that are able to destroy and dissolve foreign cells, foreign protein in form of antigens. So when cells engulf or come in contact with foreign cells, foreign protein, chemicals within the lysosome, which we call lysosomal enzymes, are released to destroy those foreign substances. And therefore, a cell is considered to be autophage, ability to destroy other organelles. So when cells have reached their lifespan, you know, cells have a limited number of years they can live. Only few cells in the body are permanent. Example given are the nerve cells. Nerve cells are permanent cells. Okay. Same with, to some extent, cardiac cells as well. Now, for example, red blood cells. When red blood cell has reached 120 days, it begins a process of death, which we call apoptosis, programmed cell death. How is that achieved? Lysosomes are involved also. Lysosomes can destroy organelles of the same cell they are found in. So, for example, here, these are lysosomes. When this cell has lived its lifespan, for example, if it's a red blood cell, it has reached 120 days. Usually it's 90 to 120 days. By 90, already they are old, and they should be removed from circulation. So lysosomes will produce enzymes that will start destroying other organelles like mitochondria, eat up the rough endoplasmic reticulum, also destroy the nucleus, and that cell will die. So that is known as autophage. Auto meaning self, self-eating. Okay, then if it's digesting foreign cells, we call it phagocytosis. Autophage is self-eating. Then phagocytosis is now destroying other foreign cells. So lysosomes are involved in both processes, phagocytosis and apoptosis. Let's look at proteosomes. Now, proteosomes do not have membrane, just like ribosomes. Now, proteosomes equally have enzymes. Proteosomes have enzymes that help in the process of apoptosis. These enzymes destroy old protein and foreign protein. So we can see them here. Proteosomes. This diagram doesn't have proteosome, only has peroxisomes. Okay. Anyway, they are involved in apoptosis, programmed cell death. Okay, so they destroy old protein and foreign protein by the type of enzymes that they have, proteosomic enzymes. So we are done with organelles. Remember, the rest of the organelles have membrane. Only ribosomes and proteosomes do not have a membrane around them. The rest are membrane bound. Let's look at inclusion bodies. Now, inclusion bodies are not organelles, but these substances listed here, 1, 2, 3, are always part and parcel of the cell. So we can't ignore them. So they fall under formed elements. So you can see these small spots, dark spots like that. Dark, 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 dark. So inclusion bodies are of 3 types. Inclusion bodies can be glycogen granules. Remember, cells need energy, and energy can only be gotten from carbohydrates. So the cell knows that at some point, the supply of carbohydrates may be erratic, and therefore stores carbohydrate in form of glycogen. And those can be seen in the cytoplasm as glycogen granules, and we refer to them as inclusion bodies, not organelles. Other inclusion bodies include lipid droplets. Lipid also is useful for energy production. So in cases where there's little supply of carbohydrates, lipids are broken down to produce ATP. Apart from that, these lipid droplets, they give color or pigmentation to cells. So if you find the cells will show a different color from one another because of the type of lipid granules or droplets they have in their cytoplasm. Example given, you see that maybe fat cells will appear yellowish or whitish in color, different from muscle cells that will appear reddish in color. But all that appearance or brownish, muscles will appear brownish, is because of the lipid droplets, which we're now calling here as inclusion bodies. Last but not the least is crystals. Cells will have some crystal. Now, crystal is because of sodium. Sodium is salt, and salt has a tendency to crystallize, to form more like stones when they mix with lipids and other steroids. So the older the cell gets, the more crystals you see in their cytoplasm, and the size of those crystals increase. So you can tell a young cell from an old cell by looking at the size and the number of crystals it has in the cytoplasm. The younger it is, the larger and high number of crystals. The younger it is, the less in terms of number and size of crystals it will have in the cytoplasm. That's all about the cellular organelles. What about the cytoskeleton, the last part of the cell we discussed? We said cytoplasm is divided into 3 parts. Cytosol, which is fluid part or aqueous environment, then organelles, then cytoskeleton. So let's quickly look at cytoskeleton. Cytoskeleton are of 3 types. Cytoskeleton is either filament, which we call microfilament, intermediate filament, and microtubule. So there are 3 protein fibers found within the cell, which we call cytoskeleton. Let's go back to this diagram here. You can see these yellow fibers. These are protein in nature. All cytoskeleton is protein in nature and produced within the body by the ribosomes, within the cell, sorry, by the ribosomes. So these are the skeleton of the cell. And we know that skeleton is more like a framework. So all cytoskeleton work to make sure that the shape of the cell is maintained. And that's why muscles have different shapes, nerves have different shapes, liver cells have different shapes, and so on and so forth. So these are the structures we're discussing. Now there are 3 types. We have the microfilaments, microtubules here. These are microfilaments, these are microtubules. You see that these are thin, the filaments are very thin. Then microtubules are thicker, like tubular in nature. So let's look at them one by one. We'll start with the microtubule. Now, microtubule is shown here. You see that they are more like tubes. Tubes. So these tubes are arranged nicely to form 3 structures important for cell function. Number 1 is centriole. So centriole are essential for cell division. Cells cannot divide until their chromosomes are separated. So like we said there, I was saying chromatin is as good as chromosomes, and we only see chromosomes when the cell is about to divide. So each cell should carry equal number of chromosomes. So who separates those chromosomes? So that this side we have equal, this side we have equal, are the centrioles. And what makes centrioles are microtubules. So what we are seeing here are microtubules. And then if you put these microtubules in one bundle, they form what we call a centriole. What is the function of the centriole? Is to separate chromosomes during cell division, without which cell division will not occur. And that is why nerve cells do not multiply in number, because they don't have centrioles. Okay, and that's why we call them as permanent cells. What other structure is formed by microtubule? Is basal bodies. Now, basal bodies, I'll come to this basal body. Let's keep it on hold for now. Let's discuss flagella and cilia. Flagella is the tail of the sperm cell. That's number one. So the sperm cell is the only human cell that has flagella. You find flagella in protozoa, rickettsia, and bacteria. Other somatic cells in the body do not have flagella. It's only the sperm cell that has flagella. But we expect cilia in some of the cells within the body. For example, cells found in the trachea, cells found in the bronchioles have cilia. Cells found in the fallopian tubes, some cells in the uterus, some cells in the epididymis there in the testes have cilia. And that cilia helps to move substances in those regions I've mentioned. For example, in the trachea, it helps to move mucus from the lungs towards the mouth so that we can spit that mucus or sometimes swallow it, because in the stomach we have acid to destroy any bacteria trapped by mucus. So flagella and cilia are attached to the cell membrane by basal body. So I'm coming back now to basal body. Basal body act as point of attachment of cilia and flagella. And that way, basal body working together with cilia and flagella, you find that flagella will help the sperm cell to move, cilia will help mucus to move towards the mouth, as it were. That's all about microtubule. Now I look at the microfilaments. Microfilaments are of two types. We have thin and thick microfilaments, or what you can say, actin and myosin. Actin is thin microfilament. Myosin is thick microfilaments. Now, all cells have microfilaments, whether it is a nerve cell, liver cell, cells of the pancreas, they will have microfilaments because the shape of those cells needs to be maintained. Now, muscle cells, to be specific, cardiac muscle, smooth muscle, skeletal muscles, you find that the whole cytoplasm here will be filled by these filaments. Yes, the cell will have organelles, mitochondria, nucleus, whatever. But in addition to that, the cytoplasm is filled with microfilament, which is thick and thin filaments, myosin and actin. And that way, it is only muscle cells in the body that have the ability to contract and relax because of presence of actin and myosin, which other cells have, but only in little amounts. Then quickly we look at intermediate filament. Intermediate filament, which is the last type of cytoskeleton. Here, we've discussed microfilament or thin filaments. We have also described microtubule. Now, we are remaining with intermediate filaments. Now, Intermediate filaments are essential in cell-to-cell attachment. So this is one cell. Let's imagine that there's another cell this side. And these cells need to remain firmly together so they can give us a solid tissue, and that tissue can give us a solid organ. To hold these cells in position to one another, you need intermediate filaments. So intermediate filaments act, apart from maintaining the shape of the cell, they act as intercellular connections to hold cells firmly together in one tissue to form organs. So that's all about the cell. So here we have diagrams. So under intermediate filaments, there are these filaments, namely keratin. You've heard of keratin, no? Nails. Nails are made up of keratin. Hair is made up of keratin. All those are intermediate filaments. Neurofilaments, these are found in nerve cells. Then laminins are found in all other type of cells. So these cells remain attached to each other. So in general, cytoskeleton maintains, number one, the shape of the cell. Then in addition, there are these different types of cytoskeleton, like microtubule giving us centrioles, flagella, and cilia. Microfilaments giving us actin and myosin. Then intermediate filament giving us keratin, neurofilaments, and laminins. Then here are summaries of which I expect you to go through and ask questions where necessary. For further studies, you can check this book of histology known as Janquira's Basic Histology. Any edition, beginning with 3rd edition going up. So you can read 3rd edition, 14, 15, going up. This is better for all information dealing with the cell and tissues. Then you can also check out this book, Witter's Functional Histology. But I recommend that at least download this book on PDF drive. It's there. Just few bundles you'll be okay. So this is where we end for today.